We've talked about carbohydrates, lipids, and proteins, and in this video we turn our attention to the nucleic acids, DNA and RNA. As the blueprint of life used to store genetic information by all cells, obviously DNA is an incredibly important class of biomolecule. RNA is copied from DNA and allows the information it encodes to be expressed as a useful cellular product. As we'll see, RNA has a number of different functions in cells. But whether we're talking about DNA or RNA, we'll see the same recurring theme of a close relationship between the biochemical composition of these biomolecules, in other words, their structures, and their chemical and physical properties, and the functions that they carry out. We can generalize the functions of nucleic acids as all falling under the general heading of information storage, transmission, expression, and regulation. Now, when we say information, we mean genetic information. Deoxyribonucleic acid, or DNA, is used by all cells to store genetic information. It's the blueprint for life, containing the instructions for how to build proteins and functional RNA molecules that carry out all cellular processes. This information isn't used directly in the cell, but is first copied into a more transient ribonucleic acid molecule, or RNA. And when we look at the functions of RNA, we see more diversity than for DNA, but the functions all relate in some way to ensuring the expression of the instructions in DNA, or otherwise regulating that expression somehow. We'll just get a brief intro to those functions in this lecture, but we'll look at them in more detail later on in the course. Most genetic instructions in DNA encode cellular proteins. As we discussed in the last video, the specific gene sequence determines the specific amino acid sequence of the encoded protein, and therefore its structure and the function it carries out in the cell. The RNA copy of those genes is referred to as messenger RNA or mRNA, and these are decoded or translated at the ribosome to synthesize the correct protein sequence. But other genes don't encode protein. Rather, they are copied to produce RNA molecules that carry out their cellular functions as RNA. In other words, they don't get translated. They are referred to as non-coding or functional RNAs. Among this class are transfer RNA, which is responsible for transporting the correct amino acids to the ribosome during protein synthesis, and ribosomal RNA, which is a part of the ribosome itself, along with dozens of proteins. Small nuclear and small nucleolar RNAs regulate the processing of mRNA and rRNA through a process called splicing, which again we'll examine in more detail later. And microRNA is an incredibly interesting type of RNA that can regulate the expression of genes either by interacting directly with DNA or with the mRNA copied from DNA, and can determine whether proteins get synthesized or not. Again, later in the course we'll look at these processes in a lot more detail, but I just want to give you a sense of how diverse RNA molecules can be and the important functions they're associated with, including sometimes serving as enzymes. When we talked about proteins, I told you that nearly all enzymes are proteins, but we now know that some RNA molecules can be catalysts as well. Specific subtypes of these ribosomal RNA, snRNA, and SNO-RNAs all function as enzymes. Because they're RNA and not protein, they're referred to as ribozymes. Both DNA and RNA are polymers of the same type of monomeric subunits, namely nucleotides. Here on the left we see the structure of a nucleotide, which consists of a 5-carbon sugar covalently bonded at this position to a nitrogenous base, which is going to be one of these 5 specific types over here. Adenine, guanine, which are both purines consisting of 2 fused rings, and cytosine, uracil, or thymine, the pyrimidines with only 1 ring. And these are symbolized by the 1-letter abbreviations A, G, C, U, and T. The sugar is also covalently linked to 1 or more phosphates. The base and the sugar together are referred to as a nucleoside, and depending on how many phosphate groups are present, it'll be a nucleoside monophosphate, diphosphate, or triphosphate. In biology, we tend to be a bit sloppy with our nomenclature, and we'll refer to the whole nucleotide by the name of its base. For example, using the term adenine, for the nucleotide that contains adenine, but strictly speaking that's not correct, and we should really call it adenosine mono-di or triphosphate. If and when you take OChem, your instructor might get upset with you if you do that, but it works for us. Now, depending on whether we're talking about DNA or RNA, we will see some differences in the nucleotides. The sugar in RNA is ribose, whereas the sugar in DNA is deoxyribose. They only differ from one another at this position in the sugar, the 2' carbon, which contains a hydroxyl group in ribose, but just a hydrogen atom in deoxyribose, hence the name deoxy. Just an FYI on the numbering here, by convention, the ring constituents of the base are numbered first. For example, here in cytosine, the ring nitrogen is 1, then 2, 3, 4, 5, 6. And the carbons of the sugar are given the prime designation to distinguish them from the atoms in the base. So 1', 2', 3', 4', and 5'. This is just so you know where that prime comes from. You definitely don't need to know the numbering of the bases, but you should be familiar with the numbering on the sugar, for reasons which will soon become apparent. The other difference that we'll see between DNA and RNA is in the bases. DNA nucleotides contain adenine, guanine, cytosine, and thymine, whereas in RNA, uracil is present instead of thymine. So in RNA, U takes the place of T. When we're talking about DNA and RNA, we're talking about polymers, chains of nucleotides linked to one another through covalent bonds. But nucleotides do have other functions in cells besides being the monomeric subunits of nucleic acid. We've already talked about one that's incredibly important in cells, ATP, adenosine triphosphate, which is a ribonucleotide. And as we've discussed before, it's the central energy transfer molecule used in cells. Another example is the coenzyme nicotinamide adenine dinucleotide, or NAD, which is an important partner in the catalysis of lots of oxidation-reduction reactions in cells, including those used to oxidize things like glucose to obtain energy. So nucleotides are important for other functions, but we're going to focus most of our attention on their role as the monomeric subunits of DNA and RNA, and we'll see how their biochemical makeup is responsible for the structures and properties of these nucleic acids. Just like we saw with amino acids and proteins, when nucleotides are joined together covalently to build the polymer, the nucleic acid or polynucleotide, the interacting functional groups are always going to be the same. In this case, the interaction is going to be between one nucleotide's phosphate group and the other nucleotide's sugar. So to get our orientation with the numbering again, starting with the nucleotide at the top, here's carbon 1', 2', 3', 4', and 5'. The same in the next nucleotide. And in this nucleotide that's getting ready to be covalently added to the chain. The substrate for synthesis is going to be a nucleoside triphosphate with 3 phosphate groups. And the new bond is going to be formed between the hydroxyl on the 3' carbon of the preceding sugar and the first phosphate group, which is attached to the 5' carbon of the incoming nucleotide. These other 2 phosphates are released as a pyrophosphate molecule. So here on the right we can see the new linkage as it's been formed. This is sometimes referred to as a phosphodiester bond because that's the specific name of this sort of covalent linkage where we have an oxygen and then a carbon. So phospho is the phosphate, di meaning 2, and ester referring to the oxygen then carbon linkage on either side of the phosphate connecting it to the 2 sugars. So just like in linked amino acids we had that N to C polarity along the polypeptide chain, here we're going to have a 5' to 3' polarity or directionality along the polynucleotide chain. Now it's important to recognize that when we say polarity here, we're not talking about dipoles, but rather we're talking about directionality along the chain. So a nucleic acid is synthesized in the 5' to 3' direction, with new nucleotides always being added onto the 3' end. The fact that the substrate for this synthesis is a nucleoside triphosphate is significant. Remember our discussion of how ATP is used in cells? A phosphate is hydrolyzed, which releases energy, and the cell links that energy-releasing reaction to some other process that requires energy. Well, the same concept is at play here. Cleaving this pyrophosphate group off is an energy-releasing reaction, and that reaction is linked to the energy-requiring reaction of synthesizing this new covalent bond. So in other words, the substrate nucleotide supplies the energy to make this reaction happen. This synthesis is not going to happen on its own. This is going to be catalyzed enzymatically, and DNA polymerase is the specific enzyme that would synthesize DNA using deoxyribonucleotides. If this were RNA being synthesized from ribonucleotides, it would be a different enzyme, RNA polymerase, but the basic mechanism of synthesis would be the same. When we look at the structure of DNA, what we see is not one polynucleotide strand, but two polynucleotide strands associating with one another to form the characteristic double helix we're so familiar with. Here's a flattened two-dimensional representation of two DNA nucleic acid chains, which makes the interaction between the two strands very clear. Here are the covalently linked backbones of the nucleotides with alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and here are the bases. They're not participating in the covalent interactions along the chain. Rather, they're responsible for the association of the two nucleic acid strands with one another by virtue of hydrogen bonding between the bases. These base pairing interactions are not random. They're specifically going to occur between adenine and thymine and guanine and cytosine, forming characteristic AT and GC base pairs. The other thing that we notice when we look at how the two strands are arranged is that they have opposite polarity with respect to one another. This strand is running in the 5' to 3' direction from top to bottom, and the other strand is running 5' to 3' from bottom to top. And that's always going to be true. When nucleic acids hydrogen bond with one another, they're always going to be anti-parallel in orientation. These AT and GC interactions are sometimes called Watson-Crick base pairs, after James Watson and Francis Crick, who correctly solved the structure of DNA in 1953. So what we think of as a DNA molecule is really two DNA molecules hydrogen bonded to one another. Due to these interactions and the strong attraction of the sugar-phosphate backbone, remember phosphates are negatively charged, with the aqueous environment, in 3 dimensions the DNA double helix folds up as shown here, with the sugar-phosphate backbone along the outside of the helical coil, and the nitrogenous bases hydrogen bonding with one another in the interior. We should also note that the bases are flat planar structures. We're seeing them here end on, and they are stacked one on top of another in the interior of the helix and are close enough that transient dipole interactions can occur as well. In other words, van der Waals interactions. As we've talked about before, individually hydrogen bonds and van der Waals interactions are weak intermolecular forces. However, in a molecule like DNA, we're talking about millions of linked nucleotides interacting in this way along the chain. So these weak forces can confer a lot of structural stability to this very important molecule. You have another video to watch that describes how Watson and Crick came to make their discovery and all the other scientists' work that they were drawing on, so I won't go into that here. But you really can't overstate just how important this discovery was, because knowing the structure explained so much about the biochemical basis for the function of DNA in information storage and transmission. Scientists had been trying to figure out the mechanism by which genetic information is passed with high fidelity from generation to generation. And by solving the structure of DNA, it immediately became apparent how this occurs. I'd encourage you to read the Watson-Crick paper. It's really short, just 1 page. It describes the double helix structure, its physical dimensions, and the base pairing interactions. And here at the end of the paper, you'll find this sentence that makes it clear that Watson and Crick knew that they had not only solved the puzzle of DNA structure, but in doing so had solved the larger puzzle of how a molecule could store and transfer genetic information. In other words, how DNA could be responsible for inheritance. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. What do they mean by that? Well, what they meant is that because the association of bases occurs in characteristic fashion, with A always hydrogen bonding with T, and G always paired with C, if you know the sequence of nucleotides in one strand of DNA, you automatically know the other. So let's say you have a cell, and its DNA double helix has this sequence in the two strands. Again, we can see the bases are paired in characteristic AT-GC fashion. Now, if that cell is going to reproduce to create new cells, job number one is to create 2 copies of the instruction manual, the DNA, to pass on to the 2 new cells, a process called DNA replication. So how is this accomplished? Well, very easily. These 2 nucleic acid strands just need to be peeled apart from one another. Remember, they're not covalently bonded to one another, only hydrogen bonded. And each strand can then serve as a template for the synthesis of the opposite complementary strand of nucleic acid. And what we end up with is 2 DNA double helices, each consisting of 1 original parent DNA molecule and 1 newly synthesized strand paired with it, being passed on to these 2 new daughter cells. So DNA replication is semi-conservative in nature, with the old template strands being conserved as half of each new double helix. This process is high fidelity, but mistakes can occur in the copying process, which also explains how new genetic variation can arise through mutations, another characteristic of genetic inheritance that needed to be explained and was explained by the Watson-Crick model. We'll look at DNA replication and mechanisms of mutation and DNA repair later on in the course in more detail. So here's another look at the Watson-Crick structure. This molecule in the middle, what's referred to as bDNA. It's a right-handed helix composed of 2 nucleic acid molecules associating by hydrogen bonding interactions of their nitrogenous bases, which are tucked away to the interior. And the sugar phosphate backbones of the 2 strands run along the outside of the helix. The helix has a constant diameter of 2 nanometers along the helical axis, and 10 nucleotide base pairs per turn of the helix. We can make out 2 distinct indentations in the shape of the helix, one shallow and one wider and deeper. These are called the minor groove and major groove. And when proteins interact with or bind to DNA, they're typically interacting very closely in one or the other groove. We should recognize that other alternate helical structures for nucleic acids have been characterized in the lab and confirmed to exist in cells. These are referred to as the A-type helix here on the left, which is also a right-handed helix but a bit more squatty, with more nucleotides per turn than bDNA. This is what a helix formed by one strand of DNA and an RNA strand might look like. So this is what we'd see when DNA is being copied to make RNA, for example. zDNA is shown here on the right, and it has a very bizarre structure. It's actually a left-handed helix, and the backbone has a weird staggered orientation. Obviously, these differences in structure would lead to different sorts of proteins being able to bind and interact with the DNA to affect its function. But not much is known about the physiological significance of this alternate DNA structure, so we'll leave it at that. When we talk about the DNA double helix, we're referring to this much more prevalent version, bDNA. One thing that we have to recognize is that DNA molecules in cells are giant macromolecules. Even if we're looking at a simple bacterium like E. coli, which would have only one circular DNA molecule, that DNA molecule is over 4 million base pairs in length. And if it were stretched out end to end, that DNA molecule would measure over 1 millimeter in length, which is a problem given that the entire bacterial cell is only about 1/1000th of a millimeter in length. So you've got a cell that contains a DNA molecule 1,000 times longer than the cell itself. Obviously, the DNA is going to have to be compacted and condensed to be able to pack more tightly into the cell, and this is highly regulated. In bacteria, this is accomplished by enzymes that interact with the DNA to introduce supercoils in the molecule. Imagine you're holding a rubber band in your 2 hands, and you twist that rubber band in opposite directions. Think about how it would coil up. This is exactly what happens in bacteria. Here we're looking at an E. coli cell that's been lysed or broken open. The remnants of the cell are here in the middle, and the DNA has spewed out of the cell in all directions, revealing those supercoils that are present. If we're looking at DNA in other types of cells, cells in the domains archaea, which are also single-celled microbes, or eukarya, which encompasses plants, animals, protists, and fungi, we'd see the DNA compacted and organized in a different manner. What we'd see instead in these cells is a complex of DNA and protein called chromatin. That is not only a means of compacting the incredibly long DNA molecules in organized fashion within the cell, but also a means by which to regulate the DNA and its expression. We see multiple levels of organization in chromatin. Initially, a stretch of DNA around 150 base pairs in length is wrapped twice around a complex of histone proteins to form what's called a nucleosome. This is a structural representation of what a nucleosome looks like. Here's the DNA double helix, and here in the center is the complex consisting of 8 histone proteins. This nucleosome is represented over here at the right as these little spheres. This first level of organization is sometimes called the beads on a string structure for obvious reasons. The next level of organization involves coiling of these nucleosomes into a cylindrical or solenoid structure like we see here. Additional structural proteins will interact with these 30 nanometer fibers and cause looping and folding to produce higher and higher levels of organization. We'll talk about this more at the end of the course, but cells can alter this structure in regulated fashion as a way to control whether particular genetic instructions are expressed in the cell or not. By making the chromatin structure less compact, the DNA is more accessible to the enzymes responsible for copying it to make mRNA. If the chromatin is more compacted, the DNA is less accessible and less likely to be expressed. So chromatin structure is dynamic. It is controlled and altered in response to the changing needs of the cell. This highly compacted structure of chromatin we see down here at the bottom, we really only see during cell division, when the replicated DNA molecules are being segregated to daughter cells. Mostly what we see is these forms above, more open chromatin or more tightly compacted chromatin. Now let's turn our attention to RNA. We've already talked about the nucleotide composition of RNA compared to DNA. We know that it's also going to be a polynucleotide chain of ribonucleotides linked covalently to one another. We know that RNA is a copy made from DNA. But let's talk about the different classes of RNA in a bit more detail, beginning with messenger RNA or mRNA, which are used to synthesize proteins. This image represents an overview of how protein synthesis is achieved. As we said, amino acid sequences in proteins are specified by genes, specific instructions in the DNA. The sequence of nucleotides in the gene specifies the sequence of amino acids in the protein. We'll see how that works later on in the course. So let's say that this stretch of DNA is a protein coding gene. And in order to express this protein, first the gene will be copied to synthesize an mRNA copy. Only one DNA strand will be copied to produce the mRNA. And the mechanism of copying is very, very similar to what happens during DNA replication. The specific hydrogen bond base pairing interactions are used to make a copy that is complementary to the template DNA strand being read. For example, if the gene sequence is as shown here, and for the sake of discussion, let's just say it's this bottom strand that gets copied for this particular gene, the enzyme doing the copying is going to assemble the mRNA molecule by following the same base pairing rules as for DNA, with the exception that RNA doesn't contain thymine, it contains uracil. So everywhere there would be a T if we were synthesizing DNA, there's going to be a U because we're synthesizing RNA. So for this gene sequence template, we'd get an mRNA copy with the sequence G, C, A, U, G, G, C, A, G, A, U, C, U, U, A, G, A, U, G. This single-stranded mRNA is then going to be decoded and the protein that it specifies synthesized in the ribosome, which is sort of like a factory floor where all of the required components for protein synthesis come together. The ribosome is shown here. tRNA molecules are critical for producing the protein because they transport the correct amino acids to the ribosome such that the correct protein primary structure is formed. Ribosomal RNA is critical to this process because it's actually a component of the ribosome itself. It's not shown here but these blue blobs are really made up of dozens of proteins along with several different ribosomal RNA molecules, one of which would be present here in the large subunit of the ribosome and would actually be the enzyme that catalyzes new peptide bond formation. Remember that term ribozyme that we talked about earlier? Now, where do these tRNAs and rRNAs come from? We have to realize that they are also copied from DNA. So just like we have this protein coding gene here, there would be some other gene in the genome that would encode this tRNA molecule, another gene that would encode each of the ribosomal RNA molecules in the ribosome. Just like with the mRNA molecule, these non-coding RNAs are specified by genes in the genome. They are copied as single-stranded nucleic acids. It's just that they don't encode proteins. They are functional RNA molecules. They carry out their functions as RNA. We've talked about this idea of the close connection between molecular structure and molecular function several times. So if non-coding RNAs are functionally diverse, you'd probably predict that they're structurally diverse as well, and you'd be right. Again, these non-coding RNAs are copied from one DNA strand, so they're single-stranded. But remember that they contain the same sorts of nucleotides capable of the same sorts of hydrogen bonding base pair interactions that we talked about before. But the difference here is that we're not talking about intermolecular base pairing, but rather intramolecular base pairing, in which the single-stranded RNA folds back on itself due to the interactions between complementary RNA base pairs, A pairing with U and G pairing with C, to form secondary structures like this stem loop structure on the left, or this hairpin structure. These blue lines are indicating base pairing interactions, so maybe there's a G, A, U, C, C here, pairing up with a C, U, A, G, G in this other region of the same RNA molecule. The molecule can form additional hydrogen bonding interactions that cause it to fold up even further, leading to higher order or tertiary structure. The specific secondary structures that form will depend on the sequence of nucleotides in the RNA, which in turn is determined by the gene sequence it was copied from in the DNA. So each RNA is going to have its own specific structure, and that's going to allow it to carry out its specific function in the cell. These images depict the structures of two of those transfer RNA and ribosomal RNA molecules we just talked about. Here's tRNA on the left. Again, this is a single strand of nucleic acid. Here's the 5' end of the molecule, and we can follow that all the way around to the other end, the 3' end. We can see the hydrogen bonding interactions that cause the linear polynucleotide strand to fold back on itself. Here are stem loop structures. And this is what the overall tertiary structure of a tRNA molecule would look like when it's fully folded. This tRNA is going to fit within a specific binding site within the ribosome, carrying the correct amino acid to be added to the protein being synthesized. The amino acid would be linked up here. Down here on the right is a different functional RNA molecule. This one is the 23S ribosomal RNA molecule, the ribozyme, that catalyzes the formation of each peptide bond during protein synthesis. It can accomplish this because it has a specific structure and properties that allow it to interact with the carboxyl and amino functional groups in amino acids that are being joined to catalyze the synthesis of that new covalent bond, that peptide bond. So different nucleotide sequences, different secondary and tertiary structures, therefore different functions in the cell. In this video, we reviewed the main roles of DNA and RNA in information storage, transfer, and regulation, and we've looked at their nucleotides from which they're synthesized. We talked about the structures and properties of DNA and some important classes of RNA, both coding RNA and non-coding RNA. And we've discussed how the biochemical composition of DNA and RNA and the specific hydrogen bonding interactions between complementary base pairs is important for structure and biological function. Now that we've discussed the 4 classes of biomolecules that all cells are made up of, we're ready to turn our attention to how they interact with one another to form the organized structures of cells and to carry out the biological processes occurring in those cells.